Now, what about the neural cell types? How do we make these neuron types of these regions that I mentioned that are really underrepresented? And so what one can do here is to do multiplex single-cell morphogen perturbation screens. So the idea is you think about possible morphogens that maybe might bring the tissue to a certain regional identity, and then you can actually have many of those conditions that you want to test. And the most powerful tool to read out what happened with supplying this morphogen to the tissue is to do single-cell RNA-seq. But how do you do single-cell RNA-seq on, let's say, 96 samples? You don't want to run 96 10x runs. That's extremely expensive and super labor-intensive. So what you can do is, and this is a screen that we did, you can hash the cells. You can provide them with antibodies that go to the cell surface and that have a barcoded oligo attached to them. And in this way, you can actually pool all, after dissociating all the organoids, you can pool them all and do the readout with single-cell transcriptomics of all these different conditions that you tested. And here we tested different timings, different concentration, and even combinations of morphogens. And what would they do to the organoid tissue, and what cell types can we get? Similarly, in Sergio Pasca's lab, this is a paper on bioRxiv, Amin et al., they had the same idea. They also grew organoids on plates, provided morphogens, 48 conditions, 96 organoids, and they did combinatorial barcoding-based single-cell RNA-seq, and then also got data. And so this is the example, actually, that we took. And now the question is, when we take this and map this back to the atlas, do we see now new cell types? How does this match the atlas that we have? And so this is what you can see here now. So we took this data set where all these different morphogens, so this is a sonic hedgehog agonist. We have KIR as a Wnt agonist combined with sonic hedgehog, KIR alone, BMP, FGF. So when we now integrate or map this, project this organoid screen data to our organoid reference or our primary reference, what this can help us with is annotation. We can first of all say, okay, now we map the annotation from our integrated atlas over to the screening data. So this allowed us to annotate the data. And what is now the coolest, I think, is that we can say, did we gain any cell type? Do we have the same cell type represented again, or do we have an enrichment that we see in this screen? And so the novelty of these cell states is shown here, where you can see the gained clusters. So this is already differential. It doesn't just show the mapping. It already subtracts what we saw before. And now these are the really gained clusters. And we can see that, for example, there are some midbrain dopaminergic neurons gained, some the Purkinje cells, also subpallial GABAergic neurons. So this is now a way to actually come up with protocols in a high throughput manner and find out how to make certain cell types that we haven't yet made in vitro. But how else can we use these screening data? And this is where I hand over again.
Yeah, thanks, Barbara. So let's take one more round. I hope you're still there. So the question is, I think it's fine. So the question is, now that you have these perturbation screens, like it's one way to use this atlas, just map, annotate, right? But can we maybe also go this next step? I think this old loop in systems biology that I think also Beate we put at some point in the proposal, right? We've been sort of carrying through, like, you do an experiment, but then you design your next experiment based on the model. So this experimental design idea, I think, is very, very promising. And here, our idea, is to, let's say we have some type of perturbed cells or organoids, whatever that be, sorry for this bad drawing, then you subtract maybe somehow that original cell type, you take a new one, and then you predict just computationally in silico how that perturbation would look like. Of course, that could be often very wrong, but you could rank these, and you could sort of, from all of these predictions, maybe take one of the most promising ones, and then get back, or a few of those. So instead of Barbara having to go in these 96 well blade through all conditions, maybe sort of have a very sparse sort of design and still be able to sort of with same efficiency come to these results. So this is a bit what we try to do, and this is, I think, what in the field, we've been talking just about this, so just going back to this, what in the field is called generative AI. Who has heard about generative AI? It's a popular thing, right? I mean, everyone who uses ChatGPT has talked to a computer that generates some response. This is sort of the idea. What it means mathematically is essentially that instead of just having these dots, you have distributions, okay? So you have, let's say, a distribution of answers with respect to a prompt, or you have a distribution that's being generated of images under some certain text condition. So some of you might have seen that particular picture here, the Pope in the fluffy jacket, which does not exist. He did not wear that jacket. But you ask the computer to do this. So let's state this example without sort of going algorithmically deep. But this is something that's just possible very, like 2 years ago or 1 year ago. It's really very, very exciting. So I've been going to Midjourney. Has someone used Midjourney? I think the new ChatGPT 4 is also really impressive. Just typed in, hey, can you draw me the Pope with a white fluffy jacket? So that got me a few popes. If you add now sunglasses, suddenly the Pope starts posing like that. It's really interesting. But what I now do is now subtract white fluffy jacket, and then you just get pictures of the Pope, okay? So I do this now by changing the prompt. Later, of course, we will do it with experimental condition. And now we add another one. So now the difference between these two should be white fluffy jacket, right? So let's add, instead of Pope, let's add Taylor Swift. This is what the Midjourney would generate for representation of Taylor Swift. And you get samples of Taylor Swift with a white fluffy jacket. So this is a generation thing. And this is essentially the idea we want to do, right? So we want to have a perturbation, let's say, with a drug, with morphogens, or with maybe also a particular disease case, and then we want to sort of come up with samples of these cells under this perturbation. We've been showing in the past that this is possible. Already 5 years or so ago, we had this what's called SCGen, so a generative model for single-cell expression, where essentially in this latent space, so this bottleneck that I was showing before, we sort of had a few perturbations where we sort of saw what was going on, and then we predict the next one by some linear interpolation, by this arithmetic, which you could call latent space arithmetic. Of course, in practice, this doesn't scale very well, and in many cases, these latent spaces shouldn't behave very linear, so we've been thinking how to extend that. And this is the idea here. This is work by Mo, Yuge, Carlo, and Nacho in collaboration with Anna and David from Meta AI, and actually been highlighted as one of the big results in AI 2 years ago in the State of AI report. Very happy about that. But the idea is, and I think the method itself could use all kinds of improvements, right? But the idea is we take our encoder-decoder framework, as you saw before, but now we sort of parameterize this in this latent space. And you don't need to look at the formulas in detail, but the key idea is sort of that you now know what the effects of the various perturbations are. And if you can model these, and you need to do some sort of nice things with adversarial learning, but if you know now, if you have such a model, you now can predict how new combinations would look like. You could maybe predict how new dosages should have an effect, and maybe also you could look at this space of perturbations, and you can visualize what the impact of the various perturbations are, how close they would be. And that's kind of interesting, because at some point you might not just sort of want to don't want to just go in those given morphogens, but maybe you want to come up with new ones that are close by in this perturbation space. So this is what we did here. This is a data set from Cole Trapnell, which is, I learned a new word from you today. You called this a mosaic, not a mosaic, no, this plot you had, you called it salt and pepper plot, no, mosaic plot, some nice confetti plot, yeah. So this is a confetti plot, and you see that you have all these different sort of labels for the perturbations. In this case, I think it was something like 200 drugs that were added to 3 different cell lines. You see just the single-cell RNA-seq readouts. But what we now did, we ran this model, and you can see that our predictions kind of match up quite well with the true ones, in particular versus the perturbation. And you can do this sort of systematically across all transcripts, and you see that the predicted always sort of quite nicely matches up with the true ones for different holdouts, such as this cell line with high dosages and so on. And you can see which factors play a role. In this case, would be drugs for epigenetic regulation. And you can also plot this sort of perturbation space where you see sort of some of the drugs behaving similarly, actually being sort of from similar categories. So I think doing some embedding like this for the particular morphogens under time, sort of having, let's say, a 2-dimensional resolution, I think would be one of the very clear and exciting next steps. And in that paper, so this I'll be showed also for genetic perturbations, and we're now currently sort of going towards more interpretable priors. And this is what we try to do here. So this is work that Leon did together with Stefan Günnemann, machine learning colleague here in Munich, where we've been now taking this model that I showed you before, but now add the structure of the perturbations as an additional latent space. So basically, you have one modality. We've been basically all the time talking about the transcript space, but maybe on top, what you sort of perturb could be time points. That could be different morphogens. But in our case, it could be sort of the drug structures of these various compounds. And the question is, can we, for new, like completely unseen compounds, potentially also predict in not too bad fashion what that effect would be. And we've been coming up with ways how to do this, which I know Skip, happy to discuss afterwards. But what you can now see as results is that, for this, where we just now have a single perturbation, then we can call this new thing chem-CPA, we compare it to a CPA in a baseline. We see, in this case, we trained it on low dosage drugs, we try to predict in the high dosage ones. And we see that this regularization of chemical space was really improving the prediction. In this case, higher is better. And this works for various different drugs and dosages, and this would be just on differentially expressed genes, sort of a bit more sensitive. But you can also quantify this fully, and you can, because we have now this chem-CPA, you can actually go what's called OOD, so out of distribution. And there you go completely unseen drugs that are sort of in the convex hull of the existing ones. And then you see that you actually still do prediction that's not bad. And this is, of course, promising, because at some point, if you're now a big pharma, maybe you can only sort of afford so many drugs to screen. So it would be useful maybe to then interpolate and sort of use this for experimental design. And we can do this also for the combinatorial setting. And this just came out, and we had this beautiful artist drawing, not AI generated yet, where they threw in sort of the cells into this mixer. We decompose it in this CPA model, and then the experimentalists sort of combine new set of drugs, and they're being generated here. Well, so as Outlook now, of course, what we really want to try to do here, and this is a project that we are sort of an ongoing discussion, I think it's early phase, but I think I'm very excited about this, would be to really go now into modeling this organoid protocol space. And in a recent ESIC grant that I got, we're sort of doing the same type of perturbations for drug space modeling. So this is the idea that we do there. So again, it's a generative AI thing, and I think this promise of experimental design, I think this will not just change our daily life with respect to paper writing or image generation, but I think I would really predict for everyone here in 5 years' time, you would have your little assistant at the side that would help you maybe design the next experiment in a potentially a little bit more informed fashion. I mean, at the moment, you have all this expert knowledge, but you already start looking to databases and you sort of put in your favorite marker genes. But if you have this additional support, this could potentially help you maybe design better experiments. So in this case, just sort of an example of what you could be doing, and we had been studying in this particular case obesity, and if you sort of see sample changing over time, maybe you can use this generative AI to sort of check out a few drugs that you actually haven't measured, but that you predict then their response, and you can sort of rank this prediction. You can then actually follow up with the experiment just for the top one. That's sort of the idea here. And I think this idea of building then these so-called foundation models is the next step. Has someone heard about the term foundation model yet? So if you want to use that, go home and sort of impress people, this is sort of the hottest thing at the moment, AI, which is essentially taking these language models, but now applying them to all kinds of other settings. So don't just do this for language, but do this, for example, for images, or do this for images of organoids or transcript resolutions. So you're building these foundation models is really, I think, something that we essentially copy what the GPTs of the world do for these other settings. And I think this is really promising. Eric Topol and others have just recently written Perspective, sort of for really applying this also in the clinical setting. We've been just talking about the omics case, but you could sort of try to integrate also other of these modalities into one big model, where you then sort of attach on top all these downstream applications, such as cell type prediction, mapping, perturbation prediction, that is fitting on this one fixed reference. So you don't need to retrain, you have like all these special cases. Nowadays, if you want to build a state-of-the-art chatbot, you need to have this language model in the background, otherwise you won't be state-of-the-art. I think similar for us, we also need at some point these in the back, and I really hope that we develop them academically and not in industry in our setting. All right.